this is going to be talking about resealed red blood cells. And the title is Resealed Red Cells, Blowing Up Red Blood Cells for Fun, Science, and Profit. So I'm Mark Scott, if you don't know that. I'm a senior scientist with Canadian Blood Services and a clinical professor at the University of British Columbia, the Department of Pathology. And for the few of you in the know, I'm also the founder and editor of the World Headline News, which you can go on the web and see. And you'll see a bit of the World Headline News in today's seminar. So, oops, let me come into here. And conflict of interest. I have lots of patents through Canadian Blood Services and Albany Medical College, but none of those are involved in today's talks. So I can talk freely about things that are now very ancient. So today is July 28th, and this happens to be, according to my calendar, Great Canadian Upheaval Day. And I was intrigued by that, and this is only in New Brunswick. And as an American, I figured that with my good red, white, and blue education, that this holiday immediately followed All You Can Drink Day. Because I was educated at the finest universities in the United States, including Trump University. And this is our history lesson for Canada. This is Doug and Bob McKenzie. And as you can see, there's the Great White North, which is Canada, which involves drinking lots of beer and everything else. So this was my understanding of Canada before I moved here 20 years ago. Since I've been here for 20 years, I've learned a few other things. That the Great Canadian Upheaval Day actually commemorates a relatively sad part of Canadian history, which is the British deportation campaigns, where the British in Eastern Canada, Nova Scotia, New Brunswick, etc., actually decided that they were very upset with the French Canadians who lived in that part of the country and who had originally settled that part of the country. And they actually, on July 28, 1755, deported all the French Canadians, or 75% of the French Canadians, to other parts of British North America, including New England and all the way down to Georgia in the United States. And they also put a number of people in prison or put them into forced labor. So really, it's not a holiday, as I discovered, but a day of commemoration. But this is history, and I kind of like history. Oops. And this is what my lab currently works on. We do a lot of what I like to think is translational research projects. And I'm not sure which way we really go. Do we take the human into the mouse or the mouse into the human? But we do a lot of things, including hematology, where we've been working on our stealth blood cells since about 1996 or so. And this is the idea that we could prevent or treat red blood cell aluminization in diseases like sickle cell anemia or thalassemia, or just everyday transfusions. We also do a lot of immunology, which has been probably the more prominent part of our research over the last years, where we're looking to use allogeneic white blood cells to induce tolerance in animals. We've also recently developed some microRNA-based biologics that could be used to either treat autoimmune diseases, so they induce tolerance, or we have another microRNA cocktail that actually enhances the inflammatory response and enhances the cancer response, anti-cancer response of PBMCs. And we've also done some work on building a better humanized mouse. And we also have a project in virology, where we have Dr. Scott's nose juice, which is a polymer-based formulation that you stick up in the nose that prevents viral binding and invasion. And then we have a lot of work that we've done over the years on red blood cell deformability. The majority of this work is using a technology that we call immunocamouflage, which is the covalent grafting of inert polymers to cell surfaces, and then either using those directly or using the byproducts, such as these microRNAs from that. So we do what I like to think is a lot of exciting things. And now that I've whetted your appetite for this, I'm going to tell you to ignore it all, because we're going to talk about something different. I'm going to go into some of my ancient research history that dates back into the mid to late 80s and into the 90s. So I've done a lot of different types of research over the years. As an undergraduate at university, I studied prairie dogs. I then went to Niger, West Africa as a rodent control biologist and killed gerbils there for a couple of years. I studied Giardia lamblia, which is a nice parasite, trying to prove that they could survive in the wild in muskrats. So I trapped muskrats for a summer or two. And then I did my PhD in Minnesota, working on oxidant defense in E. coli, have done malaria research and red cell research more recently. So have a lot of research. I won't go back to the beginning, which was the prairie dogs, but I will start talking about some things I started thinking about kind of in my undergraduate years. I became very concerned about free radicals. I was very much in favor of saving Abby Hoffman during the 70s. That's a little too far back, so I'll go a little bit more recently. In 1989, 1990, when I was both at Children's Hospital in Oakland, California, on a grant project in common with Mohan, I also went to Paris and worked there for a year as a visiting scientist. And in Paris, I did lots of things, including a lot of research on champagne. And mind you, this is in France, and so we're allowed to drink in the lab. It's a nice feature of France. And so I spent a lot of time pipetting champagne, but when I wasn't pipetting champagne, I was blowing up red cells. And that's what I'm really going to talk about today. Red cells are they're really amazing cells. They have no DNA, which in some ways makes them nice, but every day you make about 200 billion red cells and you destroy about 200 billion red cells. So you're always in sync. And most of the body's iron is in the form of hemoglobin. And so each red cell contains about 1 billion atoms of iron. And that means that you need an awful lot of iron every day. And so this led to some of my interest in iron that I won't be talking about today. But the red cell is very interesting. The human red cell circulates for about 120 days. It travels, a single red cell over its lifespan, travels almost 500 kilometers, and it goes through the heart really about once per minute with the average length of passage about 2.8 meters. And this translates into almost 5 centimeters per second. So it's moving along pretty good. For those of you here in Canada, that's about 17 kilometers per hour. If you're in the United States, it's about 10.6 to 11 miles per hour. So the red cell is really a very amazing cell. And it does all this without a nucleus. So it has, whatever it has at the start of its lifespan, that has to last the entire 120 days. There's no real repair capacity to the red cell. The functions of the red cell are pretty straightforward. Everybody knows that it delivers oxygen. It also removes carbon dioxide from the mitochondrial metabolism. It also regulates vascular flow by the binding and release of nitric oxide. All of these functions are done by the hemoglobin contained in the red cell. The hemoglobin is what makes the red cell red. And normal adult hemoglobin has 2 alphas and 2 beta hemoglobin chains. It's a tetramer, so an alpha 2, beta 2 tetramer. Hemoglobin itself consists of 2 components. It has the globin part, which is the protein, which is kind of shown down here, and then stuck inside the protein, but not covalently inserted, is the heme ring. And this is an important thing, that the heme is not covalently linked to the globin protein. It's kind of like a letter that you put into an envelope, but you don't seal the envelope. The letter can come out. And that turns out to be biologically important. The other thing to note is that when hemoglobin binds oxygen, the oxygen that you see here actually shuttles electrons back and forth from the oxygen to the iron. And what this does is it creates a resonance stabilized form where you have this unique combination here where sometimes oxygen looks like oxygen, sometimes it has an extra electron to it. And when it has this extra electron to it, it forms a free radical, which is called superoxide. So this turns out to be something that is important for the function of hemoglobin, but also turns out to be a source of problems under certain circumstances. Because, as I mentioned before, this heme ring is not covalently linked to hemoglobin. So it can come out, and when it comes out, it is now free iron floating around inside the cell, and you also generate the superoxide, which is a free radical and can lead to damage of the cell. So it turns out that the red cell, despite its very simple nature, it's just a bag of hemoglobin, really. It's a source of numerous life-threatening disorders. And in fact, the red cell disorders are amongst the best characterized of oxidant-mediated disorders. And these are driven by free radical reactions because this is a problem when you have iron and oxygen, or in this case, superoxide being generated. This results in damage to the cell via oxidative reactions. And the red cell has a lot of things that can go wrong with it. And these are just some general categories of wrong. There are porphyrias, where the heme biosynthesis is screwed up. And this is a free radical driven disease. You have hemoglobinopathies, where you have changes in the alpha or the beta hemoglobin chains that results in various diseases. And the sickle cell anemia, and the alpha and beta thalassemias. And I always emphasize the beta thalassemias because that's been my interest. You have defects in production, too few red cells or too many red cells. You also have hemolytic anemia. They can be in nature, they can arise from membrane disorders, mutations to spectrin or ankyrin, for example, or you could have enzymopathies. And this is another area of interest. And so I'll be about thalassemias, oxidant defense. So this is what goes on in terms of free radical biochemistry. You have a number of free radicals that can form. You can have the superoxide radical, which is O2 minus here. You have hydrogen peroxide, which is a reactive oxygen species, but it's not truly a free radical, but it is, as I like to refer to it, the messenger of death, because it has solubility like water, it has a long life, but when hydrogen peroxide sees free iron, as we show here, it forms the dreaded hydroxyl radical. The hydroxyl radical is so reactive that it will react with whatever is close to it, whether it be other proteins, lipids, DNA, carbohydrates. This is a very dangerous radical. And you can see how when you have a hemoglobin chain here, the alpha hemoglobin chain here, for example, and this iron comes out, the superoxide is generated, you have everything you need to drive what is called the Haber-Weiss reaction, or the Fenton reaction, as some people refer to it. And this is what's going on when you mix oxygen and iron. So iron plus oxygen always equals injury or death. So the other thing I want to point out, particularly prone to this, no other cell in the body has such high concentration of both oxygen and iron. And this is truly a potential combination. And in fact, it's one reason why evolution has favored red blood cells not having nuclei, because if you had DNA there that was meant to do something, these free radical reactions would be very mutagenic and would potentially cause major DNA damage. Because of this fact, red cells have a very well-defined catabolism of free radicals, and shown here schematically. You have the oxidant species that I talked about, superoxide, hydrogen peroxide, hydroxyl radical, and you have enzymes, for example, superoxide dismutase, catalase, glutathione peroxidase, are involved in modifying the free radicals down to water. So we've done a lot of work over the years on this. First, I did it in E. coli, then went to red cells. And we've looked at this, and I've done in particular work on superoxide dismutase, catalase, and glutathione. And I'll be talking today a lot about catalase and glutathione. So the funny thing about red cells are they can be surprisingly difficult to how changes to these normal red physiological reasons. If you want to get patient samples, the changes to the RBC structure and function have already occurred in vivo. So what you have a patient 80 cells. I just got a window showing up. Oops. If you, and then the cells you get have this very abnormal survivorship phenomena. So for example, in beta zero thalassemia, the most severe form of beta thalassemia, 80% of the red cells never actually make it into the circulatory system. And those few red cells that do make it into the circulatory system have a life expectancy of only about 14 days. Have potential compensatory mechanisms. For example, as I'll mention a bit, if a mouse lacks catalase, which is one of the ways to get rid of hydrogen peroxide, they tend to have elevated thion. So compensatory mechanisms are appreciated when you're talking about mutations and patient samples. So what people have really focused on are animal models. But it turns out there's a lot of interspecies differences in biology. For example, in thalassemia, you worry about murine versus human alpha globin chains and how they interact the cytosol of the cells. So animal models are very, very complicated in terms of the mutations you have to induce, the genes you have to insert, to make them mimic what's going on in the human. And I think it's always important to remember this interspecies difference. And I like to point this out in papers occasionally, however, as helpfully noted by reviewers, mice are not humans and Homo sapiens generate a lot of different responses than muscular. So we actually put this into a paper because that was one of the major reviews from said, what about how humans? So we wanted to make sure people understood that. So the question is, how do we study cells with enzymopathies or hemoglobinopathies? And the way we do it, manufacture them from normal human red blood cells. And you can actually see here a technician in my laboratory grinding out some resealed red blood cells in our laboratory using our exotic tools that we have in the lab. Now, important thing to note is that while we normally start off with normal human red blood cells, you can start off with normal red blood cells. For example, an enzyme, you might want to start off the correct enzymopathy by putting the enzyme mouse cells, any cell you can think of, any red cell you can think of, you can use. So what is osmotic lysis and resealing, and you do it? So these are the tools required. Now, I apologize for the highly complex science presented below. This has severe trauma in some individuals, so I'll go to this piecemeal. The first thing you need is some blood. The second thing you have to have is dialysis tubing, and then you also have to have a large beaker. Now, if you want to put something that you want to put, and we have done work at times high molecular weight iron isomers and other molecules. You can efficiently and trains inside the red blood cell up to 500. So it is very amenable to modifying the red blood cell. So how do you do this? This is the whole here on the screen. This is actually dates version of 1988. Some of you were not even alive in 1988. And very straightforward, simple method. And the works is that the blood, you take this blood and you put it as very thin film in dialysis tubing. Dialysis tubing has a molecular weight cutoff of about 3,500 Daltons. And you make sure it's a thin film because you want a high surface to volume ratio. You then take this dialysis tubing, put it in a beaker containing hypotonic buffer, And you let that mix for about 60 minutes at 4 degrees centigrade with stirring motion. So this dialysis tubing is just floating around in this beaker of hypotonic lysis buffer. Pull the dialysis tubing out and you put it into another beaker that's about 6 degrees. That is full isotonic or slightly hypotonic or tonic buffer. Let seal. And what's amazing is that you'll see the red cells actually reseal before you. So a nice deep red here. When the cells are lysing inside the hypotonic solution, you can actually see through dialysis tubing. They just become transparent almost. And then when you put it back to solution, the cells reseal and it becomes more opaque. Then you get the blood out, you take it out of the dialysis tubing, you wash it 5 to 7 times in buffer, and you have, voila, some normal red blood cells. So this is actually the mechanism that's going on inside the red cell. So you have your blood cells, you add your extracellular something here by alpha chains, for example. When you put that into solution, what happens, red cells don't, when they lyse, they don't fracture. When they lyse, there's a few more pores in the red cell, and red cell kind of collapses upon itself, and it spews its gut into the local environment. So hemoglobin inside comes out, it mixes alpha chain here. This into the isotonic solution, the red cell is kind of like memory foam. The cytoskeletal structure of the red cell is very, it remembers its shape it's supposed to be. It knows it's supposed to be a biconcave disc, and when you put it into that isotonic solution, it swells back up, and in the process of that, it sucks back up what had been outside the cell inside the cell. So it brings the alpha chains inside the cells. And then when you've washed it, the lysis pore heals itself. It's self-healing. And you now have a red blood cell that has alpha chains inside of it, or what else you've put inside of it. And this is, if you compare the characteristics of this donor red cells that you start off with the control resealed cells afterwards, you can see that these red cells are very, very similar to each other and well within the normal range for human red blood cells. And so if we look at mean cell volume, normal red blood cell in this case was about 90. Here we're about 86, but you can see a little bit larger standard deviation. Mean cell hemoglobin is normal. Mean cell hemoglobin concentration is normal. Superoxide, so if we talk about oxidant systems, superoxide dismutase enzyme is normal. The catalase active normal in the resealed red cells. Glutathione levels sound small, but within the normal range, and see that ATP levels are fine. And so these cells look normal. If you look at the microscope slide, they're biconcave discs. If you take scanning EMs of them, they're biconcave discs, et cetera, et cetera. And this is actually from a paper we published in 1990. These are very, very normal cells. And this is what happens when you're trying to load something in. So in this paper, we were loading the cells with reduced glutathione. Reduced glutathione is quite small, but you can see that there's a nice correlation between the amount of exogenous glutathione and the increase in the intracellular glutathione that we see in the cells. It's a very linear progression. And we've done lots of things with this. We've done glutathione, which is very small. We've done alpha chains, which are about 16,000 Daltons. We've done copper zinc SOD, a little over 32,000 Daltons. G6PD, G6 enzyme, which is about 59,000. Catalase, which is about 250,000 Daltons, and 70,000. We've done loading studies with all of these, and they all show very similar loading kinetics to the glutathione. So it's a very efficient way of putting things inside the red blood cell. We've done a lot of work on this. One thing we want to do is to make sure that the cellular deformability of the red blood cell was unaffected, because deformability is a key factor in determining whether or not a red cell can circulate normally inside of an animal. And so we used a technique called ectocytometry. And what ectocytometry does is it takes the red blood cells, you put them into a viscous solution, and then you spin it. And then a laser detects whether or not the red cell can form an elongated shape here, kind of like an American football. And this the laser detection measures mean red cell elongation, and it does this over an osmotic gradient. And this is just a result shown here. You don't really need to know much about this except to know that line 1 is the original donor red cell. Line 2 are the red cells after osmotic lysis and resealing. And this line 2 falls very well within the normal range for human red cells. So these cells have very normal deformability profiles. They have normal hemoglobin levels, they have normal ATP levels, et cetera, et cetera. And more importantly, these cells have normal in vivo survival. So if we do this to murine red cells and we put controlled resealed red cells back into a mouse, these cells have normal in vivo survival, which is about 50 days in a mouse. So these are very, very normal cells. So it allows us to do a lot of different things and to investigate different issues. So what we want to do was to investigate how investigate the oxidant defense system in enzymopathies and hemoglobinopathies. So one of my real interests was examining the hydrogen peroxide catabolism and the role of catalase versus glutathione, which is shown down in here. So you make hydrogen peroxide, and then you have to get rid of that hydrogen peroxide, because if that sees free iron, that leads to cell injury and death. So we actually have in the red blood cell 2 ways of getting rid of hydrogen peroxide. We have the glutathione peroxidase pathway, which converts it to water, and we have the catalase pathway, which takes hydrogen peroxide and makes water and oxygen. So we have these 2 competing approaches, and there's been lots of debates as to what's the more important approach, catalase or glutathione. And we'll talk about that. The other thing that we wanted to do was to find out how unpaired alpha hemoglobin chains actually cause the damage to the red blood cell and what they actually do to the red blood cells. We wanted to follow the sequence of events that occurred within the red blood cell. So we'll start off with GSH versus catalase. Historically, glutathione has been viewed as the most important antioxidant, as crucial in preventing oxidant injury by catabolizing endogenous and exogenous hydrogen peroxide. So the idea was that if you're exposed to hydrogen peroxide, either by eating a food or taking a drug that generates small amounts of hydrogen peroxide, that glutathione was absolutely essential to get rid of that glutathione, that hydrogen peroxide. And there's some evidence to support this. There's a disease called G6PD deficiency, and G6PD is right in here. And what G6PD does is it produces a reduced NADPH. So NADPH is very important because NADPH takes oxidized glutathione and makes it back to reduced glutathione. So this is the way this glutathione is cycled through this NADPH dependent pathway. And in G6PD deficiency, G6PD deficient red cells exhibit severe oxidant injury when exposed to hydrogen peroxide generating agents. So the classical symptom of case of this is somebody who has G6PD deficiency eating fava beans, which is a type of bean that produces, as part of its metabolism, a compound that ends up resulting in free radical production and hydrogen peroxide production. And individuals with G6PD deficiency that eat fava beans will have red cell lysis occurring in their bloodstream. So people said, aha, GSH is essential for getting rid of hydrogen peroxide. And so historically, it's been viewed as the most important way of doing it. But there are some reasons to doubt the above. For example, there are sheep populations that naturally lack reduced glutathione. And yet, if you take these red cells out of these sheep and expose them to hydrogen peroxide, there's no real evidence of oxidative injury to the red cells. So the red cells seem to be just fine, despite the fact that they lack glutathione. We also have some other conditions in both mice and in humans where you lack the enzyme catalase. You lack this enzyme here. And this is a very rare condition in humans. And when something is very, very rare, like acatalecemia is in human, it tends to imply that there's something very bad about the mutation. If you take out acatalecemic red blood cells, these cells show elevated injury when exposed to even minute amounts of hydrogen peroxide. So no catalase, and you get exposed to hydrogen peroxide, you have a lot of damage. So this was information that suggested that we maybe had things reversed, that maybe hydrogen catalase was much more important than a lot of people wanted to admit to. So we did a lot of work using human blood as well as mouse blood that we manipulated both the catalase levels in and the hydrogen peroxide levels in. And I'll just go through here. So this is catalase in this column, and this is glutathione in this column. And in human red cells, we could take, for example, human red cells that had 154,000 units of catalase activity. We could supplement that and get to the point where we had almost 500,000 units of activity. So a nice threefold increase. If we look at our acatalecemic mice that we were using, so these are mice that catalase is missing from their red blood cells, so they have virtually no activity. You can see that a normal mouse has about 50,000 units and an acatalecemic mouse has 27 units. So we actually went through and we took these acatalecemic mouse red cells and we inserted into the red blood cells bovine catalase. And what we saw was about a 15,000-fold increase in their catalase level. So we actually made these red cells in this, as I'm showing here, have huge amount of catalase activities. So we also did some other things that we looked at where we manipulated glutathione levels. I do want to point out the compensatory mechanism. Normal BALB/c mouse have about 12.8 micromoles of GSH per gram hemoglobin GSH. The acatalecemic mice have almost double that. So this is a compensatory mechanism that occurs in the acatalecemic mouse. But we also then further increased that as well. We did a bunch of experiments on these mice measuring lipid peroxidation, hemoglobin oxidation, red cell permeability, et cetera, et cetera, et cetera. And I'm not going to bore you with the graphs of that because I really want to talk about the utility of the osmotic lysis and resealing technique. But we came up with a number of important findings. First of all, the preeminent importance of catalase. And I'll give you a small hint here. Glutathione sucks as an antioxidant. And this is all done using these human and resealed mouse RBCs. So what we were able to show, catalase is clearly the primary mode of hydrogen peroxide removal in the red blood cell. We showed that entrapment of bovine catalase in acatalecemic red cells completely abrogated the oxidant sensitivity of these acatalecemic red cells. We also showed that glutathione is a very poor antioxidant, and in the presence of free iron or heme, it's a very, very potent oxidant. So glutathione is a mixed message. GSH concentration had minimal effects on oxidant sensitivity in either normal or abnormals. We could take our red cells and increase the amount of glutathione in them several fold, and it never really changed their oxidant sensitivity. We could deplete the glutathione from the red cell, and it really never changed their oxidant sensitivity. What we did show is that the oxidant sensitivity of G6PD deficient mice arises from the inactivation of catalase, as NADPH is crucial in maintaining catalase activity. So one thing that's been underappreciated for decades has been the fact that catalase also requires NADPH to keep it in an active state. catalase can turn over hydrogen peroxide, but occasionally it mishandles the hydrogen peroxide and becomes inactivated. If it has bound NADPH to it, it reverts the injury to the catalase and restores the activity to the catalase. So NADPH is essential not only to regenerate reduced glutathione, but it's also crucial to maintain catalase activity. So in essence, in G6PD deficiency, what's really happening is they have depressed levels of glutathione, but they also have depressed catalase activity, especially in the presence of oxidant generation. If you get rid of the G6PD, you then ultimately end up losing both pathways, and you will have a lot of hydrogen peroxide here that can interact with iron to produce this dreaded hydroxyl radical, which will cause cell injury and cell death. So in free radical biology, we always like to talk about it, and we talked about this in a paper John Eaton and I published several years ago, decades ago actually, which are tales of caution and woes. You can do a lot of things in science and you can try to get the answer you want. And I think this paper is really fun because we had a battle with the editor, but we finally got the prologue in here, which says that if you're not careful in science, you can come to some very bad conclusions. So on a soft and cloudless summer morning in southern Georgia, Marvin Skeeter Phelps, driving his trusty 69 Chevrolet pickup, accidentally runs over one of the largest raccoons in Macon County. 48 hours later, the dead raccoon is discovered by Clive Fairlamb, state forensic oxidologist who is looking for the dread oxidative encephalitis virus. This predates COVID, otherwise it'd be COVID. Clive's roadside brain biopsy, you crack open the skull, pull out some brain. He tested it for malonyl-dialdehyde, which is a marker of oxidative damage. And he alerted the CDC that an epidemic of OED was underway in Macon County. Scientists come in from the CDC, investigate, and eventually after 8 months discover a tire print on the raccoon suggesting that vehicle rather than virus was the cause of death. Unfortunately for Clive, for initiating this, he lost his job as the state forensic oxidologist. So 3 morals to the story: dead meat oxidizes. So something can die of something else, but it oxidizes. It does not mean that oxidation was the cause of death. So if you have dead material which is oxidized, it does not mean it did not die of oxidation. And you should always be looking for other causes of cell injury. For example, in this case, the 69 Chevrolet pickup. So with regards to oxidant defense, if GSH isn't a good antioxidant, what does GSH do? This is Clive in his later years. He had a job in Washington for a while, but that didn't turn out too well for much of America either. So I've always referred to glutathione as the Maginot Line of oxidant defense. The Maginot Line was an ill-fated French defensive line built in the 1930s to prevent future German invasions. And if you know your history, you know that the Germans had no problems conquering France in 1940, and the Maginot Line was a failure. So, but what does glutathione do? My view is that glutathione serves a crucial role as a metabolically cheap and fast-acting cellular homeostatic thermostat. It's a thermostat for the cell that gives an idea, a very real-time idea of where the cell is at. Does it need energy? Does it not need energy? Are things changing? So for example, we know it's an important cofactor, obviously, for glutathione as transferases. It plays a role in maintaining the reduced sulfhydryl groups on membrane proteins. For some of these proteins, if you oxidize the sulfhydryl group, this is a signal for the protein to be proteolyzed. So you have to maintain those in a reduced state. It also is the GSH-GSSG ratio is also an important regulator of cellular metabolism, for example, RBC glycolysis. So GSH does serve a purpose, but its main purpose is not to be an antioxidant. It's to be a thermostat for the cell, a homeostatic thermostat for the cell. So this brings us to the last part, which is going to be hemoglobinopathies. Mutations of alpha and beta hemoglobin genes are extraordinarily common. And in some parts of the world, mutations are the norm because of the pressures exerted on the population by malaria. Some of these that you know about are sickle cell anemia and beta thalassemia. My interest has been in thalassemia. The beta thalassemias, which are defects in beta hemoglobin chain synthesis, which results in unpaired alpha hemoglobin chains. So you have a lot of unpaired alpha hemoglobin chains. Alpha thalassemias are a defect in alpha hemoglobin chain synthesis and increased levels of tetrameric beta hemoglobin chains. One thing that's worth pointing out, because it becomes important, is alpha chains are inherently unstable proteins. And in fact, they're so unstable that we have, over the course of time, humans have actually had a duplication event of the alpha chains, and we have actually alpha chains. So if we talk about beta thalassemia, this unstable nature of these alpha chains becomes very important. So this is what we talked about. I showed you this before, how we put alpha chains in, and we know that the unpaired alpha chains mediate the damage, but it hadn't been possible to clearly define these unpaired alpha chains mediated these pathological. Hence, we made these model beta thalassemia cells. Right after preparing these cells, okay, 0, these red bloods, normal structure and function, they have no oxidized hemoglobin. We made these cells, and they're very normal when we start off with them. Okay, and in fact, this is just a table comparing true beta thalassemic cells with our model beta thalassemia cells, and we go through, and these are a very good model for true beta thalassemic cells, and we could follow the changes over time. And one thing I just want to go back, because I'm on this, crusade against glutathione, is I just want to show you that this becomes a real problem in beta thalassemic cells. This line here represents purified alpha chains, and what we're measuring is the auto-oxidation of alpha chains over time. And so this is where they go from being oxy-alpha chains to being met alpha chains. So they form met hemoglobin, produce superoxide, and this is oxidation and a sign of injury. And so purified alpha chains, over several minutes of time, they have some oxidation that occurs, but it's very, very slow. So these are purified alpha chains with no glutathione present. These are the same purified alpha chains if we add in some glutathione. And you can see that they go from this to this. And so the presence of iron and glutathione is a very potent oxidizing effect on alpha chains. And this is also seen in red blood cells hemolysates, which have hemoglobin, glutathione, as well as antioxidant enzymes. And if we add iron to these cells, you also get a massive oxidation there. So glutathione is a potent driver of oxidation. And we eventually came up with this model of what was going on inside of the thalassemic red cells. So the important thing to take out of this is that iron and glutathione generate a self-propagating and self-amplifying oxidative reaction. So once it gets going, it increases very rapidly and results in massive damage to the cell, as shown by all of these events over here. So this is what's driving the damage. And so a lot of things that go on. And the lessons of a lot of this research was that glutathione is bad in thalassemic cells. If we increase the glutathione levels of model beta thalassemia cells, the injury is much more severe and occurs faster. If we decrease or deplete glutathione in these model beta thalassemia cells, it actually protects the cells from injury. So glutathione can be a very potent oxidant, and in the wrong environment, i.e. with free iron around, it is a very dangerous thing to have. And this allowed us to go through all sorts of other things that talk not just about at the red cell level, what was going on there, but also some of the systemic effects that went down. So for example, we did some work where we showed that if phagocytes engulf damaged red cells, it basically gives the phagocyte indigestion, and they are very poor at processing and presenting antigen. And this can underlie some of the immunological abnormalities that are seen in thalassemia patients. So this is basic science. It's okay. Can I make a profit? Well, I'm not making a profit, but there is a company in France called Eritech that was founded in 2004, and they are using, clinically using resealed red blood cells as an adjuvant in cancer therapy. And this is a proprietary platform that they claim, and they insert drugs into red blood cells via osmotic lysis and resealing. Their technology is being used as a second-line therapeutic in the treatment of cancer. What I will point out to you is that this technique that they're using, this osmotic lysis and resealing, is no different ultimately than what we published in our paper here. But the only thing that's probably added to this is they've come up with some GMP manufacturing criteria, so they were able to get a patent on it based on the process, the aggregate process versus the general concept. So they have a lot of claimed benefits for their resealed red cells. They have prolonged duration of activity. So you put a drug into a red blood cell and it lasts for days. You know, they say 1 month versus hours. So it's days versus hours for the presence of the drug. They decrease risk of side effect. If a toxic drug is put inside of a red blood cell, it's less likely to exert profound toxicity elsewhere in the body. Very high reproducibility. You can do this technique consistently and you get a good consistent product out. It's also very fast. They say they can get it from their lab to the hospital within 24 hours, and broad applicability. You can do a lot of things with it. So this is one of their early clinical trials. This is a phase 2 clinical trial on pancreatic cancer. And they had standard chemotherapy. The control arm was standard chemotherapy. They had their risk-based arm, which is the standard chemotherapy plus RBC encapsulated asparaginase. And what they're showing here is, in this graph, is overall survival of the patients. And this is progression-free survival over here. But let's focus here on the overall survival. Pancreatic cancer is a horrible cancer. And you can see that in the control arm, even with therapy, that the maximum survival time in this population was 14 months with a mean 50% survival time of only about 4 months. When they treated these individuals with a couple of dosings of their red blood cells at early on in the course, you can see that it did have an improvement in the survival curves of these patients. And there was a prolongation of survival for 50% of a couple of months, which doesn't sound like a lot, but when you're talking about pancreatic cancer, that's a fairly substantial effect. And they had, you know, one patient that did quite well for a very long period of time. This is progression-free survival, so they were measuring, you know, the onset of symptoms and everything, but the one to really focus on is here. So there was a modest effect there. And so if we look at what their summary of this technology is that the results were modest but promising. And one thing to keep in mind is that pancreatic cancers are actually characterized by low vascularity. So the effect of the resealed red cells would be anticipated to be less effective in a tumor of low vascularity versus a tumor that had high vascularity. So it's possible that if you had a well vascularized tumor, that this drug would be much more effective. And I do want to point out, though, this is not the idea of using red cells for clinical purposes is not new. That a fellow researcher who I met several years ago and published with, John DeLoach, who was a very early pioneer. He's a veterinarian. He started working on this stuff in 1980. And he did all kinds of animals, bovine, equine, porcine, et cetera, red cells. And one of the first things he did was to use glutaraldehyde modified carrier erythrocytes to put methotrexate in it to treat dogs with cancer. So he had this in mind decades ago. So with that, I'll just end and say that blowing up red cells can be fun. It was very profitable scientifically for me, but not financially, shall we say. And I've had lots of graduate students over the years that have done some work in the lab, some of this on this, other things, and then a lot of really good collaborators over the years that I've enjoyed working with. So with that, I can end the presentation, and I'll stop screen sharing.